Hello students, I am going to go ahead and go through kind of the main process on how to go from DNA to RNA to then amino acids and then also coding strands versus template strands, right? So starting off here, let's go ahead and look at this piece of DNA. It is undergoing transcription. All that means is that our DNA is being used to create a strand of RNA, which will then be translated later on. But what we need to notice here is that similar to DNA replication, we actually have to unwind our DNA in order for the RNA polymerase to come in and undergo the transcription process. So we have to break those hydrogen strands in the middle. And only one strand is actively undergoing transcription. So that means we have another strand that is totally untouched. That is what we deem the coding strand of DNA, right? Whereas the strand that is currently being used to transcribed is the template strand of DNA. So if we go in a little bit deeper and look at the actual base pairing here, the coding strand is going to be very similar to the RNA strand, like I mentioned in discussion. So if I look, ATG, AUG, but we have to remember that RNA, right, replaces those T's with U's. So here, ATC, AUC. So the main idea between the coding strand of DNA, this strand right here, and the RNA strand, this one right here, is that they're the exact same except T's are replaced with U's. So if I were to continue on and write the next codon, it would be UCG. Everything is the same except T's go to U's. The template strand, on the other hand, is what I like to say is complementary to the RNA strand. That means the bases are complementary to each other. So for example, T goes to A. A would go to T, but since this is RNA, like I said, it would go to U instead. C to G, T to A once again to U, G to C. Complementary, right? That means the bases are complementary to each other. That's the main difference between coding and template strand. But let's put this into practice. So this is coming directly from your guys' worksheet. And I saw a lot of problems on this, which is kind of the main reason why I wanted to go over this in a video so you guys could see it and hopefully reference it. First off, we are asked to write down the coding, mRNA, tRNA strands, and then also the amino acid sequence given from the template strand. So this strand right here we're given is our template strand. So in order to find the coding strand of DNA, it's going to be complementary to the template strand, right? They're two strands of DNA. They're complementary to each other. So first, we have T, A, C. So we would write what's complementary. T goes to A goes to T, C goes to G, and then I can just keep going down. G, G, C, 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 G, T, A, T, A, T, A, A, C, G, T, G, C, forgive me for my handwriting, and then ATT goes to T, A, and then finally A. So we have successfully went from our template strand of DNA to our coding strand of DNA. These are complementary to each other. Now we have to go to the hard part. We want to find RNA based off of the DNA that is going to be transcribed. There's two different ways to do this. You can either use the template strand of DNA, which means we would have to find the complementary bases. I think that's a bit harder. Or we could instead use the coding strand that we just wrote down. In order to do that, we, like I said, just have to write everything the exact same except T's are replaced with U's. So A is going to stay the same. T goes to U. G is the same. C, C, G is the exact same. A is the same. U is replacing T, and I can just keep going down the entire way. U, A. A lot faster and a lot easier. You just have to remember that T's go to U's, right? T to U. Finally, we want to find our tRNA strand or the anticodon strand. This is going to be complementary to the mRNA and it carries the amino acids. So it's complementary. All we need to do is find what's complementary to the bases of the mRNA. A would go to T, but we're still in RNA, so it actually goes to U. U to A, G to C, and I can keep going down. C's to G's, G to C, A to U, U to A, U to A, C, G, A, U, U. Something you will notice is that this UAC, UAC, GGC, GGC, UAU, TAU, the tRNA strand is actually the exact same as the template strand, except all the T's are replaced with U's. So in the same way the coding strand goes to the mRNA strand and all T's are replaced with U's, The template strand can go to the tRNA strand and all T's are replaced with U's. This is because of the anti-parallel and complementary nature of DNA. All right, finally, let's go through the amino acid portion. So this is the answer that we got. We got all of this. And in order to find the amino acids, we want to read the mRNA using this genetic code chart. This can be found on the internet. On an exam, it will be given to you. You guys do not need to memorize any of these except for AUG for the start codons, and then the 3 stop codons right here are the only ones you guys need to be able to recognize. Otherwise, all the other ones are going to be given to you. So, one thing I saw a lot of students doing was using tRNA instead of mRNA for the code. You do not want to do that. We want to focus on just mRNA here, not tRNA. tRNA is what's complementary to the mRNA, not what we're actually reading. So, if we want to do this, let's start with our first codon, AUG. I go over here where it says first letter and I find A. Then I go up here where it says second letter and I look for U. That means we're going to be in this box, the AU box. Finally, I find the third letter of G, which corresponds to methionine, and that's what we would expect, right? It's always going to be the first one. We can use this and continue on. Second, CCG. C, right here, C for our second letter means we're going to be in this box. G gives us PRO. This is our correct answer, and it matches up, so I did it correctly. Perfect. And we can just continue on. AUA for our third one. ILE corresponds with ILE. Finally, our fourth one is UGC, CYS, and that corresponds to what they have, CYS. And then finally, we have UAA as the last one. UAA, you are annoying, is our stop codon, and this right here is our completed amino acid sequence, and we did that just from a template strand of DNA that was given. That is all that I have for you guys. If you have any questions on this, please make sure to email me and I can give you a more in-depth explanation.